Now that all of the hydrogen ions and electrons have been removed from glucose in the previous reactions, we can now use them to build ATP via oxidative phosphorylation. The reason it's called oxidative phosphorylation is because the transfer of an inorganic phosphate to an ADP to ATP is all powered by the electrons and hydrogen ions that were oxidized from the glucose from the previous three processes. Oxidative phosphorylation happens in the electron transport chain of the mitochondrion and eukaryotic cells that undergo aerobic cellular respiration. The electron transport chain is a series of protein complexes that help create a chemical concentration gradient in which one side carries more hydrogen ions than the other. This causes ATP synthase, the enzyme that makes ATP, to work. For this to work, you need the following inputs. First, 9 NADHs, which come from all three steps previously performed. You need 2 FADHs, which come from the citric acid cycles, and then you need ADPs and phosphates from the cell to be recharged. Finally, the reason why we breathe oxygen is we need it to help be that final electron acceptor when we're completely done with these hydrogen ions and electrons. If we didn't have oxygen, these would build up in our cells and create free radicals, which could actually cause unnecessary or harmful chemical reactions within our cells. And that's why we need oxygen. A lot is going on here, so let's work slowly. First, notice how NADH enters the cell. And let's get a little key here. White circles represent hydrogen ions. Notice there's a lot of them on this side of the membrane. There's a reason for this. We're trying to create a chemical concentration gradient based on these hydrogen ions. Any red circles you see will be electrons. The yellow circle down here represents oxygen, which helps to drive this process by pulling the electrons along. You'll notice cytochrome C and ubiquinone proteins. These are used to help pass electrons between each of the protein complexes. Finally, you have ATP synthase. ATP synthase is the enzyme that's responsible for actually building the ATP molecules by combining ADP with a phosphate. The only way for ATP synthase to work, though, is it needs energy. And so that energy comes from these hydrogen ions pumping across the plasma membrane of the mitochondrion and trying to hook up with the oxygen. The protein complex pumps hydrogen ions across the membrane and onto the other side. This creates a concentration gradient in which one side has more positively charged particles than the other. However, these protein complexes only work in a one-way fashion, so once the hydrogen ions pass through, they cannot come back in. The only way for them to do so is to pass through ATP synthase, powering this enzyme, forcing a chemical bond between ADPs and inorganic phosphate to make ATP. The electrons help power these pumps to push the hydrogens across since they're going against their concentration gradient. You're going from a low concentration of hydrogen ions on this side to high, so you need energy for that to work. The electrons power these protein complexes so they can push those hydrogen ions across. This entire process is called chemiosmosis, which is the development of a proton or hydrogen ion gradient. So to set the stage, let's make some ATP. First, hydrogen ions and electrons from NADH are removed to make NAD+. That will then head back to the previous reactions. Notice the hydrogen ions are pumped across while electrons are picked up by the Q proteins. The Q proteins head to the protein complex number 2, all the while FADH2 from the citric acid cycle is prepared to bring in more electrons. These electrons are then sent to protein complex number 3, while FADH2 heads to protein complex number 3, where it will release 2 hydrogen ions to be pumped across. Remember, these protein pumps are powered by the electrons. The electrons are then attracted to oxygen, which makes oxygen even more negatively charged than it already is. All the while, hydrogen ions are attracted to the extra negative oxygen, but can't go through unless it passes through ATP synthase. ATP synthase collects ADPs and phosphates, which bond together due to chemiosmosis of hydrogen ions moving through. In the end, the 2 hydrogen ions link up with the extra electrons to make water. This process can make up to 34 to 36 ATP per original glucose. So let's see what the outputs of oxidative phosphorylation via the electron transport chain. First, you have water, which can be used throughout the cell. It is the universal solvent found within living things. 9 NAD pluses are going to go back to the first steps. You're going to take 1 to glycolysis, you're going to take 2 to the transition step, and you're going to take 6 to the citric acid cycle. 2 FADs will go back to the citric acid cycle to pick up even more hydrogen ions and electrons. And then finally, you get 34 to 36 ATP, which are used by the cell. So this long process of harvesting hydrogen ions and electrons out of glucose to help power ATP synthase is all used to help make ATP and is finally being paid off. And please understand, this goes on all the time as long as you are alive. Up next, we're going to look at how metabolism happens without oxygen, and we're also going to see how lipids, proteins, and other carbohydrates can be used as energy sources to carry out cellular respiration.